Good morning, all. So I'll talk about my predominantly my PhD work, like what I did in my PhD. This is basically about coarse grain simulation on phase separating systems. So I studied various interactions and how these interactions help in phase separation using coarse grain simulation. So first we'll see like what is phase separating systems in biology. What generally phase separating system means is like if you have a mixture of like a few liquids or something, like it tends to phase separate, like consider like mixing oil and water. So they phase separate and become like two different distinct phases. So this kind of phenomenon is also observed in biology, like where like biomolecules tend to phase separate from the cytoplasm or the nucleoplasm. So for example, you see in left, like bodies like nucleolus, p bodies, paraspicle, these are small organelles which are found in nucleus and cytoplasm. And these organelles, they are nothing but a membraneless bound organelle, which are just bound by non-specific, like weak interactions between biomolecules. So what happens is like there are proteins which are like mixed in the cytoplasm or nucleoplasm. These tend to interact, come together and interact and form a membraneless organelle, which is, for example, a nucleolus or a superannular or like paraspical. So these organelles tend to have a function, but they don't have any membrane like endoplasmic reticulum or a Golgi body or something like that. So in left image, you can see these things. These are the examples of phase separating systems in biology. And right side is the image of a nuclear speckle. It is also an organelle found in nucleolus. Sorry, a nucleus. These green things are like nuclear speckles. So this is widely found in biology. So we were interested like how this is formed and what kind of interactions drive this phase separation. So what we do in our study is like we use phenomenological model. So what we do is like we generally have like all atom of like DNA or a protein structure. So what we do is like coarse grain these biomolecules into different styles. So here there are three kinds of models basically. So lattice particle, it means like one whole entity, like protein is considered as one bead or a one entity. And like DNA molecule, like a part of a DNA is considered as a one other like entity. So what happens here is like it is coarse grained in that level, so that we can capture things which are relevant in that uh, length scale and time scale. So next model is like bead spring model. What it means is like amino acid is for example, a amino acid or a base pair of a DNA is modeled as a one single bead, and it is connected by with a bond to the another bead, which is a base pair or a uh, even a amino acid. So this is a bead spring polymer model. LJ particles also is similar. So lattice particles means they can move only in X or Y direction. So they don't have any diagonal movement or even diagonal movement is there. Like it is like what do you call like a, stairway kind of movement and stuff. So LJ particle are like particles, like again they are coarse grained in a level where like whole protein is considered as one entity, or a DNA is also considered one entity, but they move through Brownian motion. So these are the kinds of model we generally use in our studies. And from these three models are used, and they are simulated using three different methods, like kinetic Monte Carlo or Langevin dynamics or replica exchange Langevin dynamics. So these are the methods which generally use. So using this, we want to study what is the role of non-specific interaction in the phase separation and maturation of macromolecules. What it means is like I'll say, come to it in next slide. So first we'll see what is in this problem what we wanted to see. So what we wanted to see is like there are like many proteins which are responsible for many neurodegenerative diseases like ALS and like Alzheimer's. So these are also formed because these proteins aggregate and form a uh, odd structure which disrupts the function of that protein itself. So this aggregation is led by phase separation. So first, what happens? These proteins are there. If some mutant, like for example, G156E mutant in FUS protein, will lead to the formation of protein interacting, and they like form a small phase separated, like we call it phase separated cluster. So generally, these proteins interacting together and form a one globule. They are called phase separated structure. So this phase separated structure again, they'll form many phase separated structure, form a fibril or like a aggregate. So this generally happens in mutated proteins which are responsible for diseases. So this is FUS is a ALS protein responsible in ALS disease, and alpha-synuclein is a protein responsible for Parkinson disease. So here also you can see like alpha-synuclein phase separates in given in a large concentration or even in a mutation condition. So we wanted to see like what will be the interaction of non-specific and specific interaction along with the polymer entropy for the phase separation, and how this non-specific interaction plays. I'll say what is non-specific interaction. So what we do is like we model the IDP of considering alpha-synuclein as a model. So alpha-synuclein has three regions: N-terminal region, C-terminal region, and a NAC region. So what happens in alpha-synuclein is like this NAC region forms beta fibrils. So when there are interaction between this NAC region, they tend to phase separate and form a fibril. So we modeled this into a coarse grained polymer where like each bead is like considered as like one amino acid. So we model the polymer like as a semi flexible bead spring polymer with a polymer of 80 bead length, and we had a patch of 7 bead length which is interacting as a NAC region. So this details and model are given here. So each bead is like a size of average size of an amino acid, and mass represents the average mass of amino acid, and persistence length means like how much length it will bend after. So if you have a polymer like so polymer, so after how many amino acids the polymer can bend. So for a protein, it is like after 4 beats, like after 4 amino acids, it starts bending. So if generally you can see like alpha helix structure, it forms after like every 3 beat there are interaction. So it is similar to that. So that was also used. So what is non-specific interaction and a specific interaction? What I was talking about. So interaction which are like very specific. So if NAC region interacts with other NAC region, it is called specific. Any other interaction between the domains, in between the domain, is called non-specific interaction. So we want to know like how these non-specific interaction guide phase separation or even affect the aggregation process. So that was our aim. To simulate this, we had like first simulated two alpha synuclein chains, like two semi-flexible polymers using Langevin dynamics, and LAMPS was the software used. So we simulated for like 900 nanoseconds. So since the beats are like uh, one amino acid in size, so we can our time scale we use and delta t we use are like much larger. So the time step we use here is like 30 femtosecond. So general molecular dynamics simulation is done in 2 femtoseconds because we coarse grain these beads. So we can the beads can move like very easily. Like there are like very less steric hindrances between the side chains of amino acids and stuff. So it is a coarse grain. So the movement is like allowed for a larger extent. So we have a time step for 30 femtosecond, and we could run for 9,000 nanosecond or 9 microsecond. So what we did is like we simulated a dimer of two alpha synuclein chains, and like uh, we tend to see like how these dimers meet and like how they form this non-specific dimer, and they mature to form a specific dimer or dwell away and go away. So for this we varied specific and non-specific interaction. It means like interaction between all the regions and interaction between red region. So red means like a specific interaction and non-specific interaction. So for NNS means like number of non-specific contacts, the contact between the non-specific regions, and NSP means contact between a specific region. Specific region means like red region, and non-specific means any region. So with increase in non-specific interaction, non-specific number of contacts increase. So we increased interaction between any region within the protein. So the protein coming together becomes like more plausible, and it increases with increase in interaction. It is very trivial. So uh, when we change the non-specific interaction, what happens to the specific interaction? That is what we were interested in. So when there is so see this graph, like where like for one interaction, for example, 1 kT of specific interaction. So we fixed the interaction between red region, and we varied the interaction between non-specific region. So for example, if you see in only one graph, for a very low non-specific interaction, what happens is like these two proteins will never come to each other to form a non-specific dimer and uh, mature into a what do we call it as a mature dimer. So this if this never happens, this will never happen. For this non-specific dimer to form, we need a little interaction between these two proteins, non-specific interaction between these two proteins. That is the red region. So what happens if there is more non-specific interaction? So what happens when if there is more non-specific interaction, when these two proteins come together, they tend to stay like that, and they'll never move. So you can see in this blue region, what happens is like two proteins nearby, they come, they don't get reorganized or reoriented to form a mature red interaction. So in a so what we conclude is like for a given specific interaction, varying non-specific interaction, we find that there's a narrow region where the a specific interaction is favored, like specific contact is favored. So this specific contact is something similar to formation of beta fibril. So to test this hypothesis, we simulated so we also calculated time scales involved in the dimer formation. So T dwell is like time it takes to dwell, and T mature is time it takes to mature. Dwell means like how much time they stay together. So staying together increases with non-specific interaction. It is very trivial quantity. So what happens is like if you increase non-specific interaction, they come together, and like they'll stay together. So this is straightforward. So what is T mature? Like time required to this red interaction to form. So this what happens in this case is like if two, what I said like earlier. So if the interaction is very low, they will come very less time to nearby, and if they come nearby, then they dwell and form mature interaction. So for them to come nearby, we there is an ideal middle region where the ideal interaction is 1 kbt, we call it. So that much interaction is required for two proteins to come nearby, dwell, and mature. So if there is like I non-specific interaction, what happens is like they'll get struck. Previously I talked about it, like so what happens is like if they come nearby also, what happens is like they'll get stuck and they'll never form. So then the interaction becomes like very high. The T mature will never happen. So in that case also T mature is very low. So we found this in a dimer case. So what happens in a multimer case? That is what we wanted to see. So what we did is like we simulated 30 such polymer chains, and saw like there is phase separated state and a mature multimer state. So we wanted to see like how these three states are formed. So again we calculated number of specific interactions, varying non-specific interaction. So what from the previous study, we fixed our epsilon sp to 1.2 kt, so which feels like ideal case, which forms a nice T mature well. So for 1.2 kt in blue interaction, it forms a nice well, energy well, where it says like T mature is like very high when there is low non-specific interaction, and T mature is very high again where there is high non-specific interaction, because they don't come nearby, they come and they get what aggregated, and like they stay in that non-specific dimer state itself. So what we did is like we fixed the epsilon sp, like specific interaction, and we varied the non-specific interaction. only in this study. So when the non-specific interaction is low, the polymer tend to be like freely diffusing state. So they don't come nearby. They are like freely diffusing in the simulation box. So when there is an ideal interaction of like 0.5 kt, like what happens here is like there is a mature state where like the polymer comes together and reorganizes itself into a mature state where the red interaction is formed. When there is high non-specific interaction, what happens is like the polymer comes nearby, they get phase separated, and they don't reorient. They just get stuck like a non-specific assembly, like in the previous state. So this was observed in the this polymers 30 polymer simulation too. So this in this graph we can see like a number of uh, non-specific contact. So it is the number of non-specific interaction NNS in the right side of the graph. In the left side of the graph, it is like radius of gyration of red beads. So radius of gyration says like how much close the red beads are. NNS says the number of interaction. It means like whether the polymer is nearby or not. So for a very low non-specific interaction, the red beads are far away, and there are less number of non-specific contact. It means like polymer are in freely diffusing state. When in the right side, you can see like rg of the beads decreases, and it comes to a value of 2 uh, 2 nanometers in size. And what happens is like number of non-specific contacts increase, and they also stabilize. So what happens here is like they come nearby and they stay together as a non-specific assembly. So what happens in the middle graph is very interesting. So RG of the polymer tends to decrease, and the number of non-specific contacts tend to increase. So in the blue region, you can see the phase separation starts, and in the yellow region you can see the reorientation happening. So the non-specific contact remains same because they are formed into a cluster, but the rg reduces. So it means like there are number of non-specific contact remains same, but the rg between the red beads like which are in this state reorients and becomes like a red region. So you can see the rg is still decreasing and becoming a smaller value, but the increase in non-specific contact is not that much significant. So it's just because of the reorientation happening in the uh, yellow to red region. So what we claim here is like so there is a ideal interaction which guides the polymer to form a mature interaction. Mature interaction is like something like a beta fibril formation in IDPs like alpha synuclein. So this we quantified using using radial distance of red beads and density of red beads along the from the center. So what we did is like we plotted how the red bead varies from the center of the simulation box or a cluster formed. So in a freely diffusing state, it is from the center of the box. In the mature state where the specifically the red regions are bound or non-specific assembly we call it, so we calculated it from the center of the formed cluster. So what we found is like fraction of red beads are like it is spread like radially across the box, so the value is like 0. So for a 0.5 kt interaction, like red beads are in center, so there is a peak at the uh, middle region, and there is a peak because like it is concentrated at one region. So for a 1.0 kt you can see the spread of red beads across the cluster formed. So this is also shown the density graph. So we also quantified this using other simulation where we did this replica exchange simulation where we varied the temperature of the system, and like we varied like 100 degree Celsius, we tend to form like different clusters of similar 30 polymer 30 polymer simulation, and we found like for a and these are the free energy graphs plotted for the specific interaction, which again gives the similar result where for 1 kt of non-specific interaction, so for 0.6 kt of non-specific interaction, there is a free energy well at 8 a specific interaction, which says like there is a formation of a mature state with like a red region in between the formed cluster. So this simulation also confirm the similar thing, and for 1 kT you can see like number of non-specific interactions are very low where the free energy well is formed, which says like there's a formation of cluster, but like red are not interacting, the specific interaction is like very low. So this was done using replica exchange simulation. So we also wanted to test this hypothesis, whether this hypothesis is true when if the two regions of specific beats are there. So what we did is like we split the specific region into two parts and simulated the system of 30 polymers again with same interactions as previous simulation. We found similar results where the non-specific interaction increases with non-specific contact increases with non-specific interaction, and specific contact as a ideal value of zero point 0.5 kT where the mature state is observed. And for a non-specific assembly, for a 1 kT of mm, uh, non-specific interaction, there is a non-specific assembly where the NSP is pretty low. So what we conclude is like this interaction region being in between doesn't matter. Only the interaction uh, value matters actually. So interaction strength of the region matters and not the distribution of the interaction region doesn't matter. That is what we saw. So to again test this hypothesis, we again did one other kind of simulation. So what we did is like we wanted to vary non-specific interaction by changing the number of non-specific beats. So what we did is like we varied the we introduced third beat which just interacts repulsively. Like so what happens is like they don't have any attractive interaction, they have only repulsive interaction between any other beat. So we introduced a black beat which has repulsive interaction with any other beat. So the number of black beat is varied so that the percentage of non-specific beats is affected. So what we did is like we varied percentage of non-specific interaction beat and non-specific interaction, but we fixed the specific interaction to 1.2 kT, and we observed like a similar result. So where in this box, uh, this quantity is like non-specific contacts per beat. Again, the whether it says like whether the cluster is formed or not. So we can see like at least 50% of non-specific interaction is required, interacting beat are required, and like again value of zero point 0.8 kT of non-specific interaction is required for uh, polymers to phase separate, the proteins to phase separate and form cluster. So when these clusters are formed, there are like two different uh, regimes of specific contacts observed. So here specific contacts is plotted for the same epsilon ns and n frac. N frac means the number of non-specific beats, and here epsilon ns is the non-specific interaction. So what happens here is like in this gray region, you can see number of non-specific contact is very high, like 14. The value is 14 to 8 to 14. You can see red and yellow. So it says like non-specific specific contact is more, means like red contacts are more. So mature state is observed. And if you increase the number of non-specific interacting bead and the number of non-specific interaction, the strength of non-specific interaction, we see a non-specific assembly. The proteins come together and get stuck in a non-specific assembly. So what we conclude from this study is like there is a when there is a comparable specific and non-specific interaction, protein tend to come to each other and they form an aggregate and get stuck together, and it is called a non-specific assembly, which is like this. So when there is a very weak non-specific interaction and a strong specific interaction, uh, like they randomly the region specifically interacting region or the, for example in adverse NAC region comes nearby and forms dimers. For a ideal weak non-specific interaction and a strong specific interaction, we see a mature mature state where like specifically interacting NAC regions come together form a beta sheet kind of structure. So we claim that there's a need of weak non-specific interaction for this strong specific interaction to come together. So this study states that phase separation means like the weak non-specific interaction guided phase separation helps in the aggregation process. So that is what we conclude from this study. So you have any doubt? This is, I will go to the next part. Hi, anyone have any doubt? Should I move to the next part?
Yeah, Rajesh, I think you can move to the next part. Like maybe if they have any doubts, they can put it in the chat also.
Oh, sure. Okay. So in the next part of the study, we want to see like effect of various interaction in the protein DNA cluster formation. So what happens in DNA damage is DNA is a vital inheritance unit. So what happens is like DNA gets damaged due to many factors like UV and stuff. So when DNA repair tends to happen, there are like a cluster of proteins which come together, and we call it phase separation here. So when DNA break has to be repaired, there's a need for a phase separated cluster of many proteins like PARP1 and FUS protein and ncRNA and many biomolecules, not only proteins, even biomolecules like non-coding RNA. These form a cluster to form a condensate, and they try to repair the DNA break or the damage in the DNA. So we wanted to study like how this non-specific interaction from the previous study helps in the DNA damage too. So in to the main question was like effect of various interaction between the interaction in the protein DNA cluster formation and role of DNA conformation in the formation of here. DNA conformation means the not the conformation of DNA exactly. It is the packaging of DNA. So how DNA is yeah, like in a chromatin level. So how this DNA conformation in the formation of role of DNA conformation in the formation of protein DNA cluster. I'll explain like what is the uh, conformation of DNA later. So how we modeled is like we modeled chromatin. We vaguely call it chromatin and DNA, like because it's a coarse grain model, so it doesn't have any alpha helix, double helix structure or something. So we have just a simple bead spring polymer model, and we just want to study the interaction between the protein and DNA and the phase separation and its effect. So it is basically a phenomenological model, as I explained previously. So what happens here is like we model the DNA as a polymer and like proteins as simple beads, and the interaction between the protein and DNA are varied, and we also varied the interaction between the DNA. So that this blue or the polymer interaction defines the DNA conformation, which I'll go show you in, like by the different models which I simulated. So these are the three interactions. So basically DNA interaction, it's like epsilon DD or a blue interaction. Like epsilon DP is like DNA protein interaction, blue red, and epsilon PP is protein interaction, which is red. So we simulated a polymer of like 100 bead length and a 90 protein like particles, and like each bead is like coarse grain for a 100 angstrom, and size is also mass of the bead is also like average mass of a nucleosome or a DNA repair protein. So we took we coarse grained in the level like each protein is like a DNA repair protein, and each bead is like a nucleosome. So what we did is like we simulated system of 100 bead polymer and a 90 bead protein, and we varied the interaction epsilon dp like DNA protein interaction and protein interaction. We see that when the protein interactions are very low, the proteins are freely diffusing, and 1 minus 5p shows the fraction of protein in multimeric state. It shows like whether the protein is freely diffusing or not. uh, if the value is low, it means like they are freely diffusing. If value is high, it says like they are in a multimeric state. So what happens if there is less DNA protein interaction or protein protein interaction, it tends to have like proteins in a freely diffusing state. When there is a uh, uh, middle value of epsilon dp and epsilon pp, there are proteins are liquid like, and they tend to form a cluster on DNA. When there is high interaction of epsilon dp or like epsilon pp, you can see the green region or the I region, like right side region, like the protein are solid like cluster on a uh, DNA, a DNA like polymer. So what is the takeaway from this slide is like high DNA protein interaction else in protein protein cluster formation. So whether this is necessary, that is what we wanted to see. So what we calculated is like from the next quantity is like number of protein protein contact with per- so why we calculated protein protein contact. Protein contact is what uh, talks about phase separation of this DNA protein cluster. So we wanted to see number of protein protein contact increasing with the protein protein interaction with varying DNA protein interaction along the three panels. So for a very low DNA protein interaction, what happens is like number of protein protein contact is not there. So for a high protein protein in right side of the graph, you can see number of DNA protein contacts also plotted, which says like whether DNA protein contact is so protein forms DNA protein forms interaction with DNA or not is shown the right side of the graph, and left side of the graph shows the protein protein contact. So the blue graph shows the DNA protein contact. The red graph shows the protein protein contact. Protein protein contact shows the phase separation of proteins, and uh, DNA protein contact shows the phase separation of proteins on DNA. So what is required is like a phase separation of protein on DNA is what is required. So we find a narrow regime of 1.5 kt and 1.5 kt of protein protein interaction, where there's a ideal peak in the graph where the DNA protein interaction is also high and the protein protein interaction is also high. So other that is the fully bound state. So what happens otherwise is like protein protein interaction is very low and the DNA protein interaction is very high, which is like a protein aggregate formation and like or the partially bound state. So the takeaway from this slide is like mm, uh, average value of epsilon dp and pp is like 1.5 kt, where the fully phase separate cluster of protein and DNA is observed, so which helps in the formation of protein DNA interaction. So what we did is like we wanted to see like how these proteins are like spread across the cluster. So we plotted number density of protein along the radial distance from the DNA bead. For each DNA bead, we check like how many proteins are nearby. So for very less epsilon dp, there is like no proteins around DNA. So like they are like the value is around zero. For a ideal epsilon dp, you can see there is peak in the 200 angstrom region, which says like there are peaks observed in the both the graphs, where like each DNA bead has a protein, at least one protein around it. So we plotted the peak density values in the right side of the graph, which shows that there's a for a ideal value of 1.5 2 kt or like 1.5 kt of epsilon pp, there's a peak density of DNA around protein, while this 3 kt of epsilon dp also shows a peak density. we wanted to see like what's the difference between this peak green region's peak density and the red region's peak density. So for this, we calculated displacement of probability of displacement of the proteins, so which says like what exactly is needed. So when the polymer is there, when the polymer of DNA is there, when there is a break in the DNA region, the proteins has to form a cluster on it, and they have to be a phase separated cluster on a not a aggregated structure. So if the structure is aggregated, it will be like not moving, and the proteins will never function perform their function of DNA repair. So when they are like phase separated cluster, there is movement inside, and there is movement inside, and the function of the phase separated cluster is not affected, and the proteins can perform their function. So what ideally should be there is like a displacement of protein should be high. That is what is required from the simulation. So we want to see what kind of interaction leads to this. So we plotted P delta x. The delta x means the displacement of protein, uh, and the distribution of this is plotted. And we when we did a summation of delta x less than 1000, probability of delta x less than 1000, and we call it a probability of a bound state. So it means like if they are like delta x is like less than 1000, it means like it is bound. It's more than 1000, it is like a freely diffusing state. We have a cutoff of 1000, and we plotted this, the probability of a bound state. So for a free diffusion there, you can see like probability of getting binding is like very less, like zero point 0.4. So randomly they come form an interaction and they bind with a given protein protein interaction. So liquid like brown state is what we ideally require. It means the uh, proteins are like liquid like, and they can perform their function in the DNA repair region. So this is the red region is the ideal required region. So we can see like for 1.5 kt of epsilon pp, and red graph is the epsilon dp of 1.5 kt, where the liquid like bound state is observed. Highly condensed state is like observed in the 3 kt of epsilon dp and any value of epsilon pp. So highly condensed state is observed. So this is what we wanted. So we found like liquid like bound state is observed in 1.5 kt of epsilon dp and epsilon pp. So we want to see like how the DNA conformation affects the this phase separation, or like liquid like cluster formation. So what we did is like we simulated the DNA inside a region. So in previous simulation, what happens is like DNA is a freely diffusing polymer, where like DNA can form any structure like but DNA was given an interaction of 1.0 kt. So they were like a little semi-flexible polymer, which is like a globule, not like a open chain. So what happens here is like we didn't give a any interaction between epsilon dd. We gave just a repulsive interaction between this polymer chain, and we gave a confinement where the polymer can freely diffuse now, and like but can be in the confinement only. So it can't move away from the confinement, it has to remain inside the confinement, but proteins can move inside the confinement and outside the confinement. This confinement is like somewhat similar to uh, a freely diffusing chain. So why the confinement was introduced is like otherwise the polymer opens up, the simulation size becomes very big. That's why the confinement was introduced. So we wanted to see whether if the previous simulation the DNA was closed, in the this simulation DNA is like more like a open like state. So we want to see like how this affects the system. We calculated similar quantities of average protein protein contact and DNA protein contact. The same thing was exact same thing was observed, like where the for 1.5 kt of protein protein interaction and epsilon dp of 1.5 kt DNA protein interaction, the peak is observed, the fully bound state, and fully bound state was observed. So in the next model, what we did is like uh, we introduce the DNA break. Here, DNA break is something like two bead, which has more interaction. So it's nothing like a break. It is just an interaction region which attracts protein. So we want to see like how this interaction plays a role. uh, the ideal interaction we found, like 1.5 kt of epsilon dp and pp, uh, affects if the DNA break is present. So for DNA break here means it's like a 5 kt interaction between protein and DNA, which affects the uh, phase separated cluster on DNA. So what we did is like we simulated uh, protein and DNA for like previously given interaction and introduced this region of I interaction after equilibration, and we saw like how it varies with the mm, protein distribution. So what we did is like we calculated two quantities from these simulations. So time for saturation of contact and gain in contact. So time for saturation means like how much proteins are on the time required for proteins on the DNA break regions to be saturated, and NPN is the contacts gained on the DNA break. So how many contacts are gained after the interaction of DNA break? So already protein can have so DNA can have protein on it because of the previous previous simulations, and when the DNA break is introduced after the simulation, so how many contacts are like gained. So this will say like whether the phase separation was helpful or not. So what kind of interaction will bring the what DNA protein DNA break protein to the break region very quickly. So we, from this what we need is like a NPN, the contact gain to be larger and TSAT, time to be lesser. So what we see is like when there is less DNA protein interaction, and for varying epsilon pp, we find that oh, left side of the graph is TSAT. TSAT is in red, and blue is a number of protein contact. So for a less, sorry, TSAT is in blue and NPN is in red. So number of protein uh, uh, contact gained on DNA break region like increases with increase in protein contact, but time also increases. So what we wanted is like a less time. So comparing these three graphs, so epsilon dp as a t sat of very low value, and npn of high value. So you can see for 3 kt the npn is very low, the time is relatively less, and but t npn is like very less for 3 kt. Just it is just because the proteins are like in a condensed form. They are not previously available on DNA, and the gain is also not there because they are like already solid like cluster. In epsilon dp of 1.5 kt, it is a liquid like state where the proteins are like weakly interacting, so they'll move easily along the DNA, and along the DNA and along the sorry, along the protein cluster also. So that's why there is a high gain in we call it like a contact on break region, uh, and T sat is very low. So uh, this ideal, so we compared 1.5 kt and 3 kt, where because this has no meaning because time is very high. Compare these two have comparable time, so we compared these two tsat, and we plotted like we see like they are very similar tsat, but for a 1.5 kt you can see a consistent or a small error bar which states that consistently tsat is very low. For a 1.5 kt and 3 kt of epsilon dp, there's a varying tsat which says like either it will be available or it will not be available, so it is not consistent. The error bar is very big. This error bar being small states that the saturation time always being less for 1.5 kt of epsilon pp and epsilon dp. So this is what we wanted. So summary is like protein interaction helps in the formation of cluster formation. Intermediate regime of protein-protein interaction and DNA-protein interaction forms a liquid-like cluster. In a instance like a DNA break, a liquid-like cluster helps the availability of protein on the break region instantaneously. That is what was shown in the TSAT. Yeah, that's all. Have questions?